Life is one. This fact, implicitly recognized by the use of single word to encompass objects as different as trees, mushrooms, fish, and humans, has now been established beyond doubt. Each advance in the resolving power of our tools, from the hesitant beginnings of microscopy little more than 3 centuries ago to the incisive techniques of molecular biology, has further strengthened the view that all extant living organisms are constructed of the same material, function according to the same principles, and indeed are actually related. All are descendants of a single ancestral form of life. This fact is now established thanks to the comparative sequencing of proteins and nucleic acids. These two groups of substances, which are the most important constituents of all forms of life, are entirely different chemically, but are both long chains made by the straining together of a large number of molecular units, up to several hundred for the proteins, often considerably more for the nucleic acids. Think of strings of beads of different color, of trains made of different kinds of cars hooked on end to end, or more appropriately, of very long words assembled with different letters. The beads, cars, or letters that make up proteins are called amino acids. Those that form nucleic acids are called nucleotides. Protein words are made with an alphabet of 20 kinds of amino acid letters. Nucleic acid words with an alphabet of 4 kinds of nucleotide letters. High-performance methods now exist for establishing the exact order in which the building blocks of these natural macromolecules follow each other in a given chain. These techniques allow scientists to decipher with great accuracy the sequencing of amino acids in proteins and of nucleotides in nucleic acids, the spelling of molecular words. The fine print in the Book of Life is now legible. One fact of immense importance has emerged from this newly gained molecular literacy. Organisms as different as a microbe, a corn plant, a butterfly, and a human being contain similar proteins and nucleic acids. These similarities are much closer than could possibly be accounted for on the basis of chance. They enforce the inescapable conclusion that these molecules, and therefore all organisms throughout the living world, are related to each other, derived from a common ancestor. As a simple analogy, compare the English word assembly with the French word assembly, used to convey the same notion. The words obviously did not arise independently in the two languages. They are related through a common ancestral word from which both are derived. The words are not identical because they have changed differently since they diverged from their common ancestor. The same is true for related macromolecules. Their sequences differ because they have suffered changes transmissible from generation, that is, mutations, as the various organisms that contain them have evolved after diverging from a common ancestor. This unity within diversity simplifies our task. We are trying to trace the history of life, not of lives. The common ancestor divides our itinerary into 2 parts. First, we must reconstruct the manner in which the common ancestor emerged from whatever materials were available before life appeared. Next, we must find out how all extant living organisms evolved from their common ancestor.